Lung cancer is never just gonna knock on your door. It's gonna break in, it's gonna redecorate, and it's gonna throw the worst kind of party. You know, the one that your immune system just can't stop? Hey, it's me, Bruno. Welcome back to Bruno's BioLogs, your go-to guide to what's new, what's next, and what's fascinating in the world of spatial biology. So I was reading this the other day. Lung cancer is now the leading cause of cancer deaths among women in over 25 countries. Now, why that's the case? Maybe it's pollution, maybe it's cotton candy flavored vapes, or something else entirely. Look, I'm not gonna sit here and try to solve epidemiology today. But what I am here to do is talk about how we're starting to get incredible spatial insights into how these lung tumors evolve. And this paper right here, well, it's gonna be using Xenium spatial transcriptomics to track lung cancer's every move, from the quiet beginnings to the full-blown invasiveness. So, let's dive in. The researchers were looking at 30 patient samples of lung adenocarcinoma, spanning from everything from early, barely bare tumors to ones that were actively invading the surrounding tissues. They didn't just grab the gene data and call it a day. They used Xenium to keep every transcript right where it belonged, like having a floor plan before the tumor started knocking down the walls. Now, here's where things start to get interesting. They looked at two expression markers, NKX21 and HNF4A. One's linked to your typical lung tumor identity, and the other to a more gastrointestinal-like cell state. Normally, you would expect these to mix a bit, but not this time. They're spatially separated. They don't just expand, they reinvent themselves. New layout, new look, same bad intentions. Building on that, when tumors start to lose their original identity, what researchers call becoming mucin negative, we start seeing markers like mucin 5AC and SPINK1 light up. That's a red flag. Because this kind of shift usually means cells are losing differentiation, which is jargon for this tumor is getting aggressive. But it's not just about the tumor itself. Zooming in on the immune interaction, the team looked at the expression of CXCL13 and CXCR5, a ligand receptor pair involved in organizing immune cells. And what did they find? Well, eventually the tumor stops yelling, "I'm dangerous!" and starts speaking just quietly enough to avoid getting detected. Now, this next part is quietly chaotic. They tracked how different tumor traits shift over time, things like CEF activation and invasiveness, and map how those features evolve together. This circular chart basically says, as the tumor starts pushing out the walls and remodeling, it doesn't go solo. It's actually bringing in neighbors along for the ride. So as things start to get more invasive, the fibroblasts aren't resisting, they're actually the ones picking out the paint colors and helping with breaking the ground. And speaking of tag-alongs, this figure zooms in even further to single cell resolution, showing how tumor cells and CEFs are physically co-located in invasive areas. Honestly, it's like the tumor is recruiting its own little support squad, laying down scar tissue, hiding from immune cells, and remodeling the neighborhood to keep spreading. So, what does all of this tell us? Tumors don't just grow like a mess of weeds. They evolve with intent. They follow spatial patterns, like they've hired an interior designer with a little flair for chaos. And what about the microenvironment? Well, it's definitely not just watching this happen from the couch. The fibroblasts, the immune cells, the neighbors, they're rearranging themselves too. Sometimes helping, sometimes getting in the way, sometimes just repainting everything beige and pretending like it looks good, and it definitely doesn't. What makes spatial biology so powerful is that it lets us see all of it in place. We're not just cataloging what genes are turned on, we're watching who's moving in, who's getting evicted, and what kind of weird open concept layout this tumor is trying to pull off. And that opens up all sorts of possibilities. New biomarkers, new risk signals, and maybe even treatments that aren't based on what a tumor is, but where it's going to be redecorating next. Kind of makes you wonder, right? Could we one day intercept a tumor just as it's starting to knock down the metaphorical wall between the kitchen and the immune system? Because if we can catch it mid-renovation, maybe we can shut down the project before it spreads to the whole house. So, from subtle beginnings to full-blown spatial chaos, lung tumors are anything but static. They're remodeling tissues, recruiting help, and generally ignoring every rule in the cellular design handbook. Thanks to spatial biology, we're not just studying them, we're watching the whole renovation unfold cell by cell. And if that made you pause for even a second, feel free to like, follow, or send this to someone who still thinks that tumors are just a pile of misbehaving cells. Until next time, I'm Bruno. This has been Bruno's BioLogs. Keep asking big questions, and I'll see you in the next time.